Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about fixation, which is part of the IBMS Specialist Portfolio in Cellular Pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss rationale and aims, learning outcomes, indicative content, resources, submission of evidence, Q&A ideas, evidence ideas, reflective statement, and some helpful tips to make candidates' portfolio better. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into this submodule. Rationale and aims of this submodule outline the general information and expected knowledge and competence that candidate needs to achieve when this submodule is completed. These are relevant to each submodule. For this submodule, the expectations are that the candidate will be able to have understanding of the changes cells and tissues undergo when they are removed from the body, have understanding of tissue stabilization via fixation and how different approaches can be employed to ensure optimal sample preservation. Explain and demonstrate processes employed during tissue fixation and how they can impact subsequent analysis of samples. Safely select, prepare and apply range of fixation techniques and reagents to samples received in the laboratory, have understanding of the impact of fixatives on the tissue, diagnosis, and patient management. The learning outcomes demonstrate candidates' knowledge, skills, and competence in this submodule. They also include demonstration of the understanding of scientific principles, laboratory procedures, quality assurance, and professional conduct. They allow candidate to work autonomously and solve problems independently. The first learning outcome is, describe the changes that cells and tissues undergo when they are removed from the body. Candidate needs to know about changes cells undergo when removed from the body. Does the candidate know what changes cells undergo when removed from the body? Do they know what is happening to cells and tissues when they are removed? Know how those changes would look like on a stained slide? Do they know how to minimize those changes? Do they know how fixation can affect those changes? Candidate could consider possible tissue changes like agonal changes of PM and surgical tissue, autolysis, putrefaction, liquefaction, cell swelling, membrane breakdown, nutrient deprivation, rapid breakdown of tissue due to enzyme release in high enzyme content tissue, effects of fixation on changes, vacuolation, gas formation, ionic imbalance, O2 deprivation, karyorrhexis, necrosis, karyolysis or loss of architecture. The second learning outcome is, explain the risks associated with the handling of fresh cellular pathology samples, including tissues and fluids, and discuss how these are managed in a lab environment. Candidate needs to know the risks associated with handling of fresh samples and how to manage them. Does the candidate know what are the risks associated with handling fresh samples? Do they know what PPE and safety measures are available to them? How would they personally deal with an unfixed sample? Candidate could consider risks like infection, aerosol generation, use of sharps that can cause injuries, contamination, cold-induced injuries during cryopreservation, chemical exposure, tissue fixation and preparation or spillage risk when working with fluids. Risk-reducing actions like safety hood, containment, risk assessment, decontamination, no-touch technique, waste management, spillage containment, PPE, vaccination, ventilation, training, SOPs, and so on. The third learning outcome is, describe situations when it may be necessary to delay fixation and discuss the challenges this presents and how these can be overcome. Candidate needs to be able to delay fixation when necessary. Does the candidate know why there is a need for delayed tissue fixation? Do they know what circumstances require delayed fixation? Do they know what challenges can arise due to delayed fixation? Do they know how to overcome these challenges? Candidate could consider situations like logistics, cryopreservation, genetic analysis, antigen preservation, biobanking, specimen size. Challenges like delayed fixation, cellular degradation, poor quality of staining, poor preservation of proteins and genetic material, infection risks due to fresh tissue, possible contamination, spillage risk for fixative, delays in diagnosis and treatment. Actions like risk assessment, ventilation, containment, vaccination, use of PPE, SOPs, and training. The fourth learning outcome is, describe the properties and modes of action of different fixatives used in cellular pathology, e.g. cross-linking, precipitation, oxidation, and discuss their advantages and disadvantages and the samples on which they are used. Candidate needs to know about various fixatives. Does the candidate know what fixatives can be used in histology? Do they know their properties and mode of action? Do they know their advantages and disadvantages? Candidate could consider cross-linking fixatives like formalin or glutaraldehyde, covalent bonds and methylene bridges formation, anchor proteins. Precipitating fixatives like alcohols or acetone, dehydration, coagulation. Oxidizing fixatives like osmium tetroxide, functional groups and cross-links. Compound fixatives like buons or Zenker's fixatives. Overall, their mode of action, properties, their advantages and disadvantages, sample types they are used and so on. The fifth learning outcome is, identify practical considerations required when fixing tissues, e.g. penetration rates in differing tissue types, and discuss how these can be overcome. Candidate needs to know about practical considerations of fixatives. Does the candidate know what practical consideration have to be taken into account when fixing tissue? Do they know how to overcome them? Candidate could consider temperature, volume ratio, concentration of fixative, speed of action, tissue size and type, ischemic time, depletion of fixative type used, diffusion and penetration rate, additives present in the fixative or pH. Actions like immediate fixation, sample opening or slicing to improve fixative penetration, ratio of 20:1 fixative to sample, 8 to 24-hour fixation time, room temperature fixation, gentle agitation, use of buffers and fresh reagents, and so on. The sixth learning outcome is, discuss with examples the impact of fixation on IHC staining patterns. Candidate needs to know about the impacts of fixation on IHC staining. Does the candidate know what impact fixation has on IHC staining? Do they know how those would look like on stained slide? Do they know how to overcome those? Candidate could consider underfixation causing edge to center pattern and morphological damage, overfixation causing antigen masking, or false negatives, antigen diffusion, antigen loss, background staining, weak signal, inconsistent staining intensity, impact on antigen retrieval, specific example 2, ER, PR, key 67 or lymphoid markers that can be significantly affected by fixation. The seventh learning outcome is, demonstrate monitoring of hazards associated with fixation and explain why these are necessary. Candidate needs to monitor hazards of fixation. Does the candidate know what hazards are associated with fixation? Do they know how to monitor those hazards? Do they know why it is necessary to monitor those hazards? Candidate could consider different types of fixatives, their exposure limits, monitoring of exposure, health hazards, safety hazards, environmental hazards and biological hazards associated with fixation, risk assessments, and health and safety audits to assess hazards, use of ventilation or respirators to minimize exposure. Reasons why they are necessary to monitor like employee safety, service user safety, public safety, environment safety. The eighth learning outcome is, explain why it is important for samples to be optimally preserved and discuss the impact deterioration can have on patient outcomes, management. Candidate needs to know the importance of optimal fixation. Does the candidate know, why is it important to optimally fix specimens? Do they know, what can if samples were not properly fixed? Do they know, how poor fixation can affect patient outcome or management? Candidate could consider over and under fixation, structural damage, cellular degradation, and their effects on diagnostic and therapeutic accuracy, reproducibility of results, compromised sample analysis or integrity, misinterpretation or misdiagnosis, digital pathology and AI algorithms, patient safety, delays, costs, resources, affected staining quality and so on. The ninth learning outcome is, discuss common technical issues which may arise during fixation including their root cause, and how these may be overcome without compromising sample integrity. Candidate needs to know about technical issues of fixation. Does the candidate know, what are the common issues that can arise during fixation? Know how they can affect sample integrity? Do they know, how to overcome them? Candidate could consider under and over fixation, delayed fixation, fixation pigments, or distortion of architecture, use of wrong fixative. Steps during troubleshooting like problem identification, corrective and preventive actions, communication, record keeping, and so on. Indicative content highlights background knowledge that might be assessed during the viva. They may or may not be demonstrated by the learning outcomes or candidate's evidence. In this submodule, candidate is expected to have the knowledge and understanding relevant to the deterioration and changes samples undergo once removed from the body and how this is minimized, e.g. autolysis, cooling/freezing, fixation. Situations where delayed fixation of samples may be required and why it is important that fixatives are sometimes not immediately used, e.g. frozen section, genetic analysis, biobanking. A range of different fixation techniques and how they be applied to tissue samples, e.g. heat, immersion, perfusion, microwave. A range of different chemical fixative agents, how they differ and how they can be applied to a variety of tissue samples, e.g. common fixatives e.g. formaldehyde, glutaraldehyde, specialist fixatives, buffering and pH, impact of temperature, fixation artifacts, awareness of tissue penetration time, diffusion rate. Methods to improve penetration time e.g. slicing. The health and safety considerations that apply to both the practice of sample fixation and the chemicals employed in tissue fixation. COSH. Risk assessment. Environmental monitoring. The impact of poor sample preservation on patient management and outcomes, e.g. interpretive/diagnostic issues. Impact on reflex testing e.g. IHC. Indicative content also highlights skills and competence the candidate is expected to be able to do. For this submodule, candidate must be able to safely prepare, handle, use and dispose range of FIVs commonly available within histology department. Resources section on the Brightspace contains some very useful information for this submodule. Submission of evidence section on Brightspace informs the candidate what evidence needs to be submitted for each subsection. Candidate needs to submit answers to questions that were set by the training officer. I will share some examples of possible question suggestions later on. Candidate also needs to select an additional piece of evidence relevant to this submodule. For evidence ideas, please have a look at the video for trainee. Each submodule also requires the candidate to submit their reflective statement relevant to their practice and training in each submodule. These are some example questions you could give your candidate to test their knowledge, competence, and skills about this submodule. These should be based around the learning outcomes mentioned earlier. These are some more example questions you could use for this submodule. You could ask them to complete tables you created, based on the learning outcomes. Or you could use images to test candidates' knowledge and competence. Supporting evidence should be selected by the candidate. It needs to be based around the learning outcomes and indicative content, and be relevant for each submodule. The evidence should allow the candidate to demonstrate their specialist knowledge and competence in each submodule. It should be specific and relevant to the candidate's in-house practice. If you would like to see specific ideas for supportive evidence, please have a look at the video for trainees. Each submodule also contains reflective statement, which is asking the candidate to summarize their role in the lab in the context with each submodule. How do they demonstrate that they can draw conclusion from what they have learned, reflect on their current practice and how it can change in the future due to gained knowledge? They could think about what is their lab role in context of this submodule. What impacts can their actions have on their colleagues, department as a whole, and for the patients? They also need to write a personal reflection on their training in the lab, and explain why they chose their evidence. This is very personal to their own circumstance. It should demonstrate their ability to be self-aware when analyzing gaps in their knowledge. They can use it as an opportunity to reflect on aspects of their training, application of their gained knowledge and skills, and how they have achieved their goals. They should think about what their evidence demonstrates, and why they chose this specific evidence for this submodule. Also, they should write about what their training was like when it comes to this submodule and how their practice changed due to their training. Their reflective statements can also be used as a CPD evidence in case they are chosen to be audited by the HCPC. I would also like to share with you some helpful tips that can make candidates' portfolio look really good. Ask them to try and present the information in various formats such as tables, animations, pictures, posters, flowcharts, videos, photographs, PowerPoints, diagrams, graphs, infographics, charts. This nicely breaks down a lot of text that is usually present in specialist portfolios. Ask them to try and make it a little bit more interesting to the reader, which will be, and eventually the assessor. Ask them to try and put themselves in your or the assessor's shoes. How would they like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture is worth 1,000 words. I will make a video where I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also ask them to try and use various types of evidence, not just one type, like a case study, for each submodule. They could use experiments, case studies, annotated results, troubleshooting, controls production, annotated images, risk assessments, glossary, training presentation, certificates with reflective statements, nonconformances, validations, verifications, comparisons, ECHA results, annotated audits, kappa or DATIX, Q&A. Ask them to try and bring in their personality. Ask them to think about the ways they like to learn best. If they like experimenting, let them perform experiments. If they like troubleshooting, let them perform troubleshooting. Ask them to choose that demonstrates their specialist understanding, knowledge, and skills, and how they apply this in their practice. And one more thing, ask them to keep the learning outcomes and indicative content in mind when they are selecting their evidence. Learning outcomes that require the candidate to demonstrate something would be ideal for covering with their evidence. And this brings me to the end of today's video. Hopefully you now have a better understanding about the learning requirements for this submodule. And I hope you also have a better idea what questions you can choose to test candidates' knowledge. And my final take-home message? Keep those learning outcomes in mind when selecting questions and guiding the candidate. Bye for now.